specific immune response. And so in order to look at the specific immune response, I'm going to break it into individual parts and then bring those all together so you can see how it all works together. The first component are the B lymphocytes, or B cells. These mature in the bone marrow, circulate in the blood, and then settle in lymphoid organs. After they mature, they differentiate into different cell types called plasma or antibody-producing cells and memory cells, as I'll show you in a minute. The T lymphocytes, or T cells, mature in the thymus, and they can remain in the thymus, they can circulate in the blood, or they can just reside in lymphoid tissue, like spleen, for example. Like B cells, they require antigen binding to surface receptors for activation and continuation of replication. They produce cytokines that are chemicals that have an effect on other cells. So here's just a little dichotomy of how these originate from lymphocyte stem cells, and then these primordial stem cells can then migrate to different places and mature in their different ways. So if they mature in the bone marrow, they become B cells, and stem cells that mature in the thymus become T cells. The B cells, as I said, can differentiate upon stimulation into antibody-producing plasma cells, and then memory cells, which retain a memory of the specific antigen that stimulated their production. These are typically long-lived cells that give an individual long-term immunity, long after circulating antibodies are gone, for example. And these are the reasons why we would need a booster shot, for certain diseases after 10 or whatever years. You typically run out of memory cells, and then you get a booster shot to re-stimulate the formation of memory. The plasma cells are the cells that produce antibodies to fight the current infection. Over here on the T cells, the T cells come in different varieties here, particularly the T helper cells, which are essentially like the quarterback of the immune response, which help other cells grow and differentiate by producing various cytokines that they produce. They can also suppress immune activity as well. As with the B cells, the T cells also have memory. So these, again, are the long-lived responses to specific invaders. There's also a version of T cells called the cytotoxic T cells, which actually kill intracellularly infected cells, similar to the natural killer cells that I've talked about. But these CTLs, or cytotoxic T cells, are very specific to very specific antigens that stimulated their production. Phagocytosis is an important part of the innate immune response, but it also bridges the gap between the innate immune response and the specific immune response. And so certain phagocytic cells, like monocytes and macrophages and dendritic cells and neutrophils, are responsible for ingesting antigens, or in this case viruses like SARS-CoV-2 or bacteria or whatever, killing them and then displaying the antigens on the surface to let the other components of the specific immune response know what's attacking them. So phagocytosis is mediated by certain coatings on the invading organism. So in this hypothetical example, we have a bacterial cell, in this particular case, one that has antigens on its surface, and those antigens have been bound by antibodies. And so the phagocytic cells have antibody receptors on their surface that would allow them to grab onto this guy and then ingest it and kill it. Another type of binding occurs through complement C3b, as I've already mentioned. So the bacterium would have some, or virus or whatever, would have some kind of a surface component that C3b would recognize and bind to. And then the phagocytic cell has a C3b receptor that can then bind to that, which in turn would allow it to grab onto this guy more easily, take it in, and kill it. And over here in the middle column, you see degree of binding. So antibody binding is good, C3 binding is a little bit better, but the combination of those two binding apparatuses is synergistically better than the other two. And so this other column here lists is with the heading opsinin. Opsinin, again, is just a molecule on a surface of an invading organism that enhances uptake by a phagocyte. So the opsinin in this case is the antibody. The opsinin here is the complement C3b, and then the opsinin in this panel is both the combination of antibody and C3b. So this is the ideal situation. The phagocytic cell can then grab onto these guys tightly. This guy can't get away, and it will be ingested and hopefully destroyed. So this process of antigen binding by the phagocytic cell results in antigen presentation. So here's our antigen in this particular case. Imagine it as a virus particle, and this phagocytic cell grabs onto it through one of the opsonization mechanisms I just showed. The microbe is brought into the cell and destroyed, and then pieces of the virus are displayed on MHC antigens on the surface. And so here's the MHC receptor with the piece of the virus or whatever displayed. So basically what this guy is now saying is showing this to the rest of the immune response and telling the rest of the immune response what's infecting it. The macrophage then, displaying that antigen on MHC type 1, or I'm sorry, type 2, will shake hands with different components of the immune response. In this particular case, it's shaking hands with the T cell. Two different varieties, either the CD4 T cell or a CD8 type T cell. CD4 T cells are called T helper cells, and they're, as I said, are like the quarterback of the immune response that will go on to stimulate other cells. If it happens to be a CD8 type T cell, that's the cytotoxic T cells. These guys behave like natural killer cells or NK cells, where they can go off and then fight very specific intracellularly infected cells. They can destroy them. So in this particular case, the macrophage presents the antigen on the MHC class 2. Here's the antigen right here, and it'll look for the right T cells that have the right receptor to recognize this. And so in this process, once the match is made, once the handshake is made, that stimulates the macrophage to produce some factors and the T cell to produce some factors. And in turn, that causes the macrophages to divide and as well the T cells to proliferate and activate. Some of these T cells become the T helper or the cytotoxic T cells that fight the current infection. Some of these guys become the long-lived memory cells. And so these guys provide the surveillance for long-term immunity. And so this has been one of the issues with COVID-19, people wondering whether or not we have immunity. And they're basing it on antibodies, circulating antibodies that are depleting over a short period of time, which is typical, which is normally what happens. But what people aren't talking about very much anyway, is the fact that we produce this memory response. And that is really what we need to be looking at when we're asking the question of whether or not there's longer term memory and immunity. So here's another look at this process. In this particular case, here's a virus, a SARS-CoV-2 or virus-infected cell. And these guys, this particular virus-infected cell has been has been infected and is now displaying virus antigens on its surface. In this particular case, it's using that MHC type 1, or in this cartoon it's called HLA type 1. HLA is just a specific kind of MHC that's specific to humans. So instead of calling it MHC class 1, they're calling it HLA class 1 because it's, they're saying it's specifically a human. So the human HLA1 is displaying the antigens in this particular SARS-CoV-2 infected cell. The antigen presenting cell, this guy here that has eaten some virus, is now displaying the virus antigen on MHC2 or HLA2, pictured here. And that antigen presenting cell could be a macrophage or dendritic cell or a monocyte, is now able to display these to pools of T cells, for example. And the right T cell in this pool, the one with the right receptor, will be selected for to proliferate and fight the current infection. It'll proliferate and divide into memory and active fighting cells. The appropriate cell will also produce the right cytokines that will trigger more recruitment and more activation of different kinds of immune system cells. It'll produce an antiviral response that triggers interferon production, like through the RNase L pathway, and increased natural killer cell activity and increased antibody production and so forth. These T cells will also stimulate B cells, the antibody producing cells. So this SARS-CoV-2 infected cell is displaying antigens. This guy has eaten some viruses and is also displaying antigens. So these cytotoxic T cells that have the right receptor can recognize this guy as well, and they can divide, and they can help, fight the current infection. One of the things that these guys would do is if they recognize this as a cell in distress, they can actually kill that infected cell. They produce the perforins and the granzymes and those kinds of things, in addition to producing interferon gamma and TNF alpha. These molecules, like I said, are chemoattractants and increase antiviral defenses. And so this guy will directly kill this guy. These CD8 T cells will directly kill this infected cell to prevent it from producing viruses. This antigen-presenting cell will stimulate more T cells, such as CD8 T cells and CD4 T cells, to go on to fight the infection. So the T cell will then shake hands with a B cell, the appropriate B cell, a B cell that has also experienced the antigen. So what's cool about B cells is that they are also antigen-presenting cells, meaning that they can bind to an antigen. So here's a piece of that virus. They can bind to it with receptors on their surface and take that in, display that on MHC2, and then put that out into the world for T helper cells to recognize. This reaction that you see here is similar to what I just showed you here, where the T cell and the antigen-presenting cell or macrophage are interfacing. So again, B cells are also antigen-presenting cells, and that's how they interact with T cells. Once this interaction takes place, these guys communicate with each other through cytokines. So the T helper cell that you see here produces cytokines to trigger this guy to proliferate and become memory cells for surveillance and plasma cells for antibody production. So again, the long-term memory cells and the shorter-term plasma antibody cells. And so it looks, something like this, and I've, zoomed out a little bit more. So here's our B cell. The B cell has a receptor on its surface that binds to some kind of a microbial antigen, for example, and that it'll internalize that, and then that particular B cell, in this particular case, will be able to interact with a T cell that you see here. This T cell can then shower the B cell with cytokines that would cause it to differentiate into the long-lived surveillance memory cells and the short-term plasma cells that will produce more of these antibodies to fight the current infection. So it's these antibodies that we would look for and detect in our antibody kits, looking for SARS-CoV-2, for example. But if these don't exist anymore, if they've depleted, it's easy for people to say, oh, this person's never been infected, or oh, this person no longer has immunity, which is completely wrong, because we may not, the individual may have been infected before and may have a long-lived population of memory B cells to fight the infection upon secondary stimulation. And so this shows you this process called clonal selection in B cells. So in this particular case, the particular B cell that is clonally selected to fight the infection has to be very carefully determined. And so in this particular case, imagine having a pool of B cells. Only one kind of B cell or a few kinds of B cells will have the right antigen binding site on its surface for a particular antigen. And so that particular B cell out of a pool of thousands will be the one that's selected. And this one will then recognize the appropriate T cell and then will differentiate into the right pool of memory and antibody producing plasma cells. This is called clonal selection. Again, you don't want all of these B cells to come to the fight because they won't make the right, a lot of these guys won't make the right antibodies. Only the guy that you see here that has the appropriate antigen binding sites on its surface will be able to make the appropriate antibodies to fight that particular infection. And so here's a diagram of the, again, SARS-CoV-2, some of the features of the virus, and this virus then sees ACE2 receptors on cells along your respiratory tract, for example, and the virus enters the tissue, the cells in particular, uncoats, releases its viral RNA. The virus RNA is translated into proteins that are then used to make more virus particles. More virus particles go out into the body and can infect other cells. In this process, phagocytic cells or antigen-presenting cells, like you see here, will attack these guys and gobble them up and then present antigens, in this particular case, to a T helper cell. The T helper cell, or it can be a cytotoxic T cell as well. And so if it's a cytotoxic T cell, remember that this guy will go off to kill intracellularly infected cells. So this T helper cell will go back and kill this cell, so it no longer can be a virus factory. If it's a T helper cell that interacts with this antigen-presenting cell, then this guy will shake hands with the appropriate B cell through the clonal selection process. That will stimulate the B cell then to produce plasma cells that can produce antibodies to fight the infection. But it will also stimulate this guy to produce a memory response, just as this guy will produce a memory response as well, or will differentiate rather into memory T cells and fighting T cells. So this shows you just an example of sort of the timeline of the two major kinds of antibodies that are produced by B cells, at least in the bloodstream, and also in tissue as well. But so some period of time, so imagine this is the initial infection point, and some period of time before you start to see symptoms is indicated in the so-called incubation period, the period of time in which you don't really know you've been infected. You're not showing symptoms, and this can vary depending on the virus. For SARS-CoV-2, it's probably around 14 days is the maximum, but some patients it takes longer, but who knows. But generally it's considered to be, 5 to 7 days, but usually within a 14-day window, most people will start to show symptoms. So once the incubation period is up, then the person goes into that prodromal phase or symptom phase, and usually the first symptom is fever and chills and a little soreness in the throat, in the chest. And so during this phase, your body produces this type of antibody called IgM. Not quite as specific. It's fast, rapid production, not as specific for the particular organism that stimulated its production. But over a certain period of time, then IgM will peak, plateau, and then fall off. Meanwhile, the more specific IgG antibody will start to be produced. And again, these numbers are not very, super specific for SARS-CoV-2. They're estimates. And so again, during a certain period of time, you'll have a bit of both IgM and IgG, and then eventually that will plateau and then start to drop off. And so again, this amount of time will dictate whether or not you have circulating antibodies that will show up in a antibody test for SARS-CoV-2 or for any pathogen. That if you're just looking for IgM or IgG. And that's why they show the broken lines here, because who knows? We don't know how long it takes. In my case, after I was infected in February, and I did my antibody testing in May, and so it was about 3 months or so where I had no IgM that was detectable, and I had just a faint amount of IgG that was detectable. So I'm guessing that for SARS-CoV-2, it's probably about 3 months before you start to lose circulating antibodies that are detectable enough by our kits. So then upon reinfection, because you have memory, after some amount of time, those memory cells can rapidly jump into the fight, and they don't need all that extra stimulation that I showed you before. And so then you can get an even higher and faster response, specific response of IgG. So you can see the difference in peaks here. And this y-axis is antibody level. And so again, it's a faster response, but it's also a stronger response upon secondary infection. And that's the beauty of memory in a specific immune response. And so here's a few outcomes on these COVID-19 test kits. They, looks a lot like a pregnancy test. So in this particular scenario, there's no infection or not enough detectable antibodies, meaning that the individual probably was infected, but for whatever reason didn't fight the virus with these particular IgG or IgM antibodies. Or, it was too long ago that the infection happened, and therefore the individual is testing negative. But you put your blood sample here, and the blood will move across this area here, this conjugation pad, and eventually will, if there's an antibody antigen response, that is, if your blood has antibodies for the virus, it'll pick up the antigens that are here and then settle either here if it's IgM or here if it's IgG. And then the control that you see here is a totally different reaction. But I talk about that somewhere else, so you can look at that other lecture on this. But you need to have the control band to show up positive in order for this to be useful. So in this particular case, it's negative for IgG, negative for IgM, and that tells you that it was either no infection, not enough detectable antibodies, or the infection happened too long ago. In this particular case, an individual had a relatively recent previous infection, not too recent. And the reason why we know it wasn't too recent is because the IgM is negative, the IgG is positive, and thankfully the control is positive, so we know it's reliable. So again, this individual had a previous infection, not too recent previous infection. Here's a more recent infection result. So an individual that has IgM positive but no IgG, that's a really recent infection, or an individual that still has IgM and also IgG positive, that's also a recent infection. So you can take this information and look at it with respect to this to get a sense for why you can get a general idea of how long ago the infection was, just based on whether or not IgM or IgG are positive or both. Another thing that I have to point out, is that we don't just fight infections with IgM and IgG. We also produce a third kind of antibody called IgA. IgA is more protective along mucus-coated cell types, like that along your respiratory tract and your digestive tract and the urogenital tract as well. And so our kits, these kits that I've been talking about, mostly look for IgM and IgG, which is very limited. It doesn't take into consideration that people can fight this infection with IgA as well. And so kits need to be more holistic, look for IgA as well. And so here's the virus has lots of opportunity to invade these cells because there are ACE2 receptors on these cells. And so all along this tract are these M cells that you see here, these little weird-looking things. And these M cells, their job is to take in antigens, present those to the system below, and then fight the infection at the local level as opposed to in the blood. And so the M cell will present the antigens to antigen presenting cells below that you see here, and then those antigen presenting cells will present antigens um, to B cells and T cells, and that will trigger a localized antibody response in the along the tract. And so goblet cells that you see here produce mucus, and submucosal gland. This also produces mucus to keep this coated. That's another defense barrier against virus infection, because you produce mucus, you can cough that up essentially, and then swallow it into your stomach. So you cough that up and swallow it into your stomach, or you can spit it out. And so that's another non-specific defense. But as far as specific defenses go, this localized immunity based on M cells and IgA production predominantly are important. And so that's pictured here more in a closer detail. So this M cell is basically this thing here. And so imagine here's my SARS-CoV-2 virus, and or pieces of the virus. These M cells will process that, will phagocytose those, take those in, present those to antigen presenting cells, macrophages or dendritic cells, and then those activated macrophages will then present those to T cells, and then the T cells will become activated and present that information to the B cells, which will then proliferate into antibody-producing cells. And these antibody-producing cells will produce a lot of what's called secretory IgA to fight virus infections or whatever along the mucous membrane. And so again, in a lot of people, the SARS-CoV-2 and things like that are beaten here at the level of the mucus using IgA. And so if you're just looking for IgG and IgM, you could miss all these people that are now immune to the disease because they used different mechanisms to fight the infection.